Hey everybody, we're starting the same module, but we're going into lesson 2 from last week. So we're going to jump right into this, get started, try to get this done as quick as possible so you don't have to listen to me talk too long. And you can start working on the assignments. This week's assignments, there's going to be 2 check for understanding quizzes that you have to do, and then you also have like 3 assignments. One of them is a virtual lab that looks pretty cool. That I think you'll have a little bit of fun with. So let's go ahead and jump right into this and we'll get going. So this week we're covering mitosis. Last week we kind of just introduced what cell division is, talked a little bit about stem cells and that type of stuff and differentiation. So now we're going to go into mitosis, and I'll let you read through the learning objectives here as well as on Canvas so you can get ready to kind of understand that. A little bit on some definitions. You'll need to make sure that you know what chromatids are, stem cells, we kind of talked about that last week, parent cell, mitosis, daughter cell, centromere, and chromatin. Again, this vocabulary can be found on Canvas as well. All right. So as a quick introduction, in this lesson, we're going to look closely at the process of cell division as a way to understand how complex organisms like humans and other mammals develop. It's interesting that one single cell can divide into a variety of cells, and that each of these cells have different structures and functions and shapes and so on. And we kind of saw some of that last week as well. So how does this happen? Why are our muscle tissues different from our skin tissues? Could stem cells be used to help cure injuries and diseases? And we've kind of already started talking about all of that, but we're also going to keep exploring it from this point forward and see how that kind of happens, what the mechanism is. All right. So what is division versus differentiation? Okay. Before we start looking closely at cell division, it's very important to understand that there's differences in cell division and cell differentiation. Cell division is just what it sounds like. It's the process of one cell basically becoming 2 cells, and then maybe those 2 cells divide and become 4 cells, and so on. Cell division typically includes 2 processes. The first process is called mitosis, which we'll cover. And actually, there's a couple of videos on Canvas that are really good. And then that's basically where the nucleus of the cell is being divided. That's what mitosis is. And then the second part of that is cytokinesis. And cyto is basically referring to the cytoplasm of the cell. So this is where the cell, the rest of the cell, basically splits in half. Cell differentiation, on the other hand, is a process in which cells become specialized to perform specific tasks. This is the process that gives the rise of different types of cells. A stem cell can become a nerve cell or a muscle cell or, a pancreas cell or you name it, a skin cell, any variety of cells. So we're going to be focusing mostly on mitosis and cytokinesis as we go through here. The differentiation part will come up a little bit later on. So there's basically a 2-part video on Canvas on mitosis that you should watch. The phases are going to be briefly outlined here. I'm going to kind of point out a few things real quick, but the video goes into some detail. And sometimes when you're watching, a single picture, they say a picture's worth 1,000 words. Well, then a video might be worth, 1,000,000 words. So sometimes it's good to kind of see these processes in motion and hear it from other objectives or viewpoints. All right, so the first phase when we're talking about the cell cycle is interphase. And I don't even know if you want to consider this a phase, but interphase is basically the cell being itself. It's doing its job. Most cells spend a majority of their time in interphase. This is the point in the cell where the cell is just kind of doing what it's supposed to do. If it's a muscle cell, it's busy contracting muscles and making your muscles function. If it's a nerve cell, then it's sending electrical impulses to wherever they need to go. If it's a cardiac muscle cell, then it's helping your heart beat and so on. It's just doing its job, if you will. Later on, environmental factors as well as internal factors kind of trigger the cell to start cell division. And that's where we enter these phases. Prophase is where you start to see this chromatin. If you look, you see these squiggly lines inside the nucleus and interphase. These things condense up into these things called chromosomes. That happens during prophase. Later in prophase, the nucleus breaks down. The nucleus kind of dissolves away, and these chromosomes are basically kind of starting to organize. And then you have these things called centrioles in animal cells. You don't see them in plant cells, but you see them in animal cells. They start migrating to opposite ends of the cell. Okay. Then we enter metaphase. This is where the centrioles, they have these spindle fibers that kind of connect to the center of these chromosomes, and they help organize the chromosomes right along the middle. That's kind of how I memorized metaphase. Metaphase means middle, meta means middle, and the chromosomes line up along the middle. And then you have the centromeres on each end, which are connected to these spindle fibers as well. And this is going to be the mechanism that kind of pulls these chromosomes apart. Now you got to remember during interphase, right before we divide, actually something else triggers inside the nucleus where all the DNA is copied. So each one of these chromosomes are made up of something called sister chromatids. If you look at this yellow one, for example, this large yellow one, this end and this end are basically exact copies of one another. And then what happens is after they're lined up along the middle, we enter anaphase, and that first one that I just pointed out, this exact copy of DNA and this exact copy of DNA begins to be pulled apart or separated to opposite ends of the cell. Then we start going through telophase, which is basically where the nucleus in each of the cells start to reform. The spindle fibers kind of break down. There's something called a cleavage furrow that begins to form between the cells. And eventually we enter cytokinesis, which isn't pictured, which is where this finally pinches apart. And you have two complete cells that go back to looking like maybe a slightly smaller version of the cell over here in interphase, except they're exact copies of each other. They're clones of one another. So again, I encourage you to go back and look at those videos because it kind of really describes it pretty well. All right. So what is the role of DNA and chromosomes? DNA is important, an important molecule to all living things. It's basically, as we learned a while ago, DNA is basically the information on how to build proteins, right? It's the information on how we make proteins inside our cells, and those proteins control our characteristics. So if you remember, in the previous section of this lesson, we learned that how DNA controls everything in a cell by providing the instructions for protein production. We learned that last semester. This is also true for the process of mitosis and cytokinesis. DNA provides the instructions for the processes of mitosis and cytokinesis. Before the sperm and egg cells fuse, they each contain 23 chromosomes. However, once fused, the chromosomes match up, and the 23 chromosomes are 23 pairs of chromosomes, so we end up with a total of 46 chromosomes. So you got to kind of keep some of this terminology in check. We have something called diploid and haploid. So haploid equals 1. The prefix haplo means single. So the sperm and egg cell are called gametes. They're considered haploid because they only have one single copy. And then diploid means 2. So all the other cells are called somatic cells, and they have 2 sets of these 23 chromosomes for a total of 46. And we're going to get into more of that, and I'm sorry I was in the way there, but we're going to get into more of that when we talk about meiosis, and that'll be coming up next week as well. All right. So again, still talking about the role of DNA and chromosomes. These terms are true for eukaryotic organisms, meaning that if their somatic cells have 10 paired chromosomes for a total of 20 chromosomes, then their haploid cells, the sperm and egg, would only have 10 of those, right? Because they're split. As seen in the video on canvas, the new cells created through cell division are exact copies of one another. Therefore, it's essential that each new cell gets a copy of each paired chromosome. So that's where, as I mentioned previously, in interphase, we have to make a copy of the DNA to make sure that our cells have a full complement of the DNA that are required. So this is kind of putting some color coding in here to kind of help us understand what's going on, all right? So the different shades of color illustrate different pairs of each chromosome. So in the image above, you're going to see how the chromosomes are copied and separated to ensure that each new cell gets the correct amount. And the cell pictured is from an organism that has 3 paired chromosomes, or a total of 6 chromosomes. So if we look at our original over here, we got 1, 2, 3, 4, 5, 6, and they're all slightly different colors, right? We got different colors in each one of these. And I'm going to be bouncing back and forth from this page to the next page, which is kind of going through this stepwise. We'll go through step 1, 2, 3, 4, 5, and so on, okay? So I'll be bouncing back and forth. So let's look at step 1 real quick. So at step 1, this is the cell and the nucleus before the DNA has been copied. Notice that the red chromosomes are one pair, the blue a second, and then the green a third, and then the different shades of color represent each individual of the chromosomes. So if you take a look, we have 6 chromosomes in there, right? So this would be interphase. Now, during this phase, there's a signal that triggers the cell to make a copy of each of these. So it makes a copy of the dark red and the lighter red, and a copy of this lighter blue and this kind of purplish, and this green and this kind of yellowish. So we end up with 2. So now, if you take a look, you'll see this one here now has 1, 2. The red one has 1, 2. The blue has 2. The purplish color has 2. The greenish color has 2. The yellowish color has 2. Okay? This would be where we enter prophase. Okay? Number 2 says the DNA has been copied, and that's what I was just talking about, during interphase, and now we kind of see it lined up. Number 3, the copied chromosomes now attach at the centromere and line up at the equator. So this is entering metaphase. When we go back, we're going to look at 3, which is metaphase. Notice the dark shade is attached with a dark shade and a light with a light. This helps ensure that each new cell gets the complete set of chromosomes. So if you take a look again, this would be interphase, prophase. Now we're at metaphase where they're kind of starting to line up, right? We're getting them lined up along this cell, okay? Notice that each of these are kind of lined up in conjunction with what color they have. That way we make sure that each cell gets the right set of chromosomes. There's also something here, and this isn't really talked about here too much, but there's also something here called crossover that can occur that helps add to the genetic diversity that we can see in organisms. But we'll go into more of that maybe later on. Okay. Number 4, step 4 is the chromosomes have now separated, moved to opposite ends of the nucleus. So we have the splitting of the chromosomes. So now one red is on one side, one red is on the other. So we got a yellow here, red here, this darker color here, the purple, 1 each, lighter blue, 1 each, green, 1 each. So they've kind of separated out. And then finally we got step 5, the nucleus and the cell have divided, and each new cell has a complete or the same copy of DNA, so they're identical to the picture in number 1, as the daughter cells in number 5 are genetically identical to the parent cell. So you have basically these perfect clones. All right. Now, binary fission, that's how it happens in eukaryotic cells. Binary fission is a little bit different. Prokaryotic cells like bacteria reproduce without adding any genetic diversity or sex. They do sometimes do something called conjugation, which is a primitive form of sex where they just exchange bits of DNA, but it's not sex as we kind of think of it, where we have a sperm and an egg fusing and forming, a 46 chromosome zygote. This means that the offspring are exactly like the parent. They are genetically identical. This mode of reproduction is actually called binary fission. And we can look at this image over here. This represents a bacteria cell. It has this circular kind of DNA. The DNA kind of is duplicated. Then it's split up. We split it in half, and then you end up with your new cells. So what's the purpose of cell division? What is the purpose of this? Now that we understand how division occurs, let's take a look at why it happens. Cell division is important because it allows living things to accomplish several things. And we kind of talked about this again in the previous lesson. Growth, right? We all start out as a single cell, and the cell division allows us to grow into trillions of cells and become this complex multicellular organism like we are today. We also use cell division during cell repair or tissue repair. Every time you get a paper cut, every time you get an injury or you scrape your knee, even just on a day-to-day basis, you're losing thousands and thousands of skin cells. You take a shower, you wash your body, you scrub. and those cells are lost forever while we have to replace them. So cell division is what's happening under the scab of your skin or when you get a cut. It's also happening in our skin when we're losing the outer layer of our skin, which comes off, the dead keratinized skin that comes off. It's being replaced constantly with new skin cells that are being done through cell division. And then reproduction, prokaryotic single-celled organisms reproduce using cell division, passing on their genes. We do a special type of cell division called meiosis, and we'll talk more about that next week. So what is the rate of this division? In the case of bacteria and rapidly growing organisms like a fetus or a young animal, uh, cell division takes place in a pretty rapid procedure, right? So it happens rapidly. The organism can grow and thrive. Most cells in fully grown organisms don't, they basically divide on a much slower scale. There's even some cells that don't divide at all after birth. You're born, for example, with all the nerve cells you're going to get and all the muscle cells you're going to get. Now you can get smarter by forming more connections between the nerve cells, and you can actually make your muscles larger. You're not gaining more muscle cells. You're actually just kind of increasing the thickness of the muscles that are already there. The exception to this is that there are areas like skin and bone marrow which are constantly generating new cells. If there's an injury sustained, the cells can divide and quickly heal the area. That's some cells that kind of divide quickly. This means generally as we get older, cell division rate slows for a majority of our cells. This is why typically it takes older people longer to heal from identical wounds as younger people. Remember that repair is one of the main functions of cell division. The story of cell division begins at fertilization and the formation of a zygote, a diploid cell resulting from the fusion of two haploid gametes. The need for new cells continues throughout our life, but it's greatest during the early years when we're first being developed, right? The fertilized egg divides into two cells, which then can give rise to 4, and then 8, 16, 32, 64, 128, 256, 512, and so on, until we have a fully grown adult, right? The rate of cell division is much less under normal circumstances. Cell division in an adult takes place only when singles indicate that the need to grow or reproduce cells signals that kind of trigger the need to grow or replace cells that have been damaged or worn out. Some cells, as I mentioned, don't ever divide again. They enter something called phase 0 of mitosis, where they just kind of hang out and do their job the entire time, never going through mitosis. All right. So let's cover the assignments real quick, and then we'll finish up. Do all the check for understandings. I think last week there was 1. This week there's 2. You have a science notebook assignment, which is the mitosis science notebook, assignment 2.4. Then you also have a mitosis review assignment, which is a pretty good assignment that kind of gets most of this on 1 or 2 pages, most of this information. A good thing to kind of do, and maybe even have available for some of the quizzes when we do them later. And then you have assignment 2.6. This is kind of cool. I signed myself up for this. You'll probably have to get a free, sign up for a free membership to the Gizmos lab. You can click the assignment. It'll take you there. And then it's a cool little lab on mitosis, a virtual lab that you can work on. And that should be a little bit more entertaining than obviously just kind of reading or doing research. So that's it for this week. I will talk to you later, and if you have any questions, stick around or come and see me during office hours. Don't forget, this week, Wednesday, tomorrow, there will be no office hours, and then we'll start up the Wednesday office hours again next week. All right, guys.